Hi, my name's Kryssianna, and I'm going to go ahead and talk to you all about QIIME2 today. And here are the topics that we're going to discuss: an explanation, limitations, advantages, controls, and then we're going to look at some figures. Okay, so what is QIIME2? QIIME2 is a microbiome bioinformatics platform. It provides comprehensive and reproducible microbiome data science. Its sequences can be taken from a sequencer such as the Illumina, which we learned about in class, and those sequence files can be imported into the program. So the most common raw data comes from the high throughput amplicon sequencing, which can have single end or paired end DNA sequence reads in either a multiplex or demultiplex format. So in order for QIIME2 to locate these files, though, a manifest file must be created by the user. It contains two columns, and one that lists the name of each sample, and the second that has the file path. So once this QIIME2, once the QIIME2 has all of the data imported, it will then create artifact files, which is just a way for it to translate the data that you imported there into a way that it understands it. And then you're free to do your analysis and create any visualizations from those analyses. What are some limitations to this program? Because we're inputting large amounts of data, they might not all be coming from the same protocol. So that variation could change the data that you have collected. Your data could be, part of it could be incomplete, part of it could be incompatible because of this protocol variation. So you may not be having all of the same standard data, and that could cause some errors in the analysis that you do complete. Another big issue is some of the plugins require a subject matter expert. So you might not be there. It's not always a plugin and play. You might need to have some sort of person on your team who has that specification, who can understand, like, oh, this sequence is biologically important to be conserved. No, it is not an error. We need to keep it in there. So what are some of the advantages? There are many open source software programs, but QIIME2 is not only open source and free, it's always being upgraded. It's always having plugins added to it. It is able to generate interactive visualizations, and it has multiple user interfaces. So whether you're a data scientist that prefers command line code, or you are new to computational analysis and you are more comfortable with the graphical interface, it allows both of those types of people to be able to be using this program. So that's really allows this program to be a collaborative space. And then also it can detect corrupted results, so it does know when your results cannot be reproduced, which is kind of an interesting feature to have, to let you know, to let you be aware of that. Okay, so what are some of the controls to this program? The most important is to be able to filter out low quality reads, and there are two program commands that do this, DA2 and dblur. The DA2 is the more common choice, and it will remove sequences from that were created in error. Okay, so here are some figures from the first paper. The first paper was written by the individuals who did program QIIME2. So you'll see that there are actually quite a lot of different figures over here. And it's important to note that these figures are not all coming from the same data set. I think their intent here was to show how many different visual, interactive graphics that could be created through the use of this program, and that's why I also chose this picture, because I did want to show that variation. So that first figure you'll see is actually a principal component analysis plot. The second is a taxonomic bar chart. You'll see a line graph there in the middle that is determining the diet of infants, whether it's through breast milk or formula. Then you'll see a normal bar chart, and over here, this silhouette of the man is actually quite an interesting way for them to depict where these samples are coming from or being generated on a person. So it really gives you a different way, a multidimensional way of looking at this data. And on this next slide, this figure came from a paper where they were doing diverse microbiome data in a comparative study. This is a quality plot, and for this plot, they went ahead and zoomed in on position 33. Basically this, if they zoom in on a position, it will randomly generate a sample of 10,000 out of the sequences, and then you can see what those normalized sequences are on this chart. Okay, and this last figure is from a paper on oral microbiota, which is actually really cool. I wish that I was able to show you what it would look like if I were to download this data in the file format that QIIME2 can read it in. I would take it to the website QIIME2 View, and then I would just drag and drop this this graphic in there, and then I would be able to look at this data in real time. So I know that this picture actually doesn't really give you a lot of information, so it's hard for you to know what it's actually talking about. But if I were to do that, you'd be able to see, okay, what are the red dots looking, what are they identifying? And I could go through and I could zoom in, zoom out. I could look at some sort of specific oral microbiota, all the different categories, and it's actually really cool. So I would highly encourage you all to go through that site. And actually, they have a few examples on there. You don't need to have the data. You could also pull it from papers and just kind of copy and paste it over by saving the file to your local computer and then dragging and dropping it into there. And it'll let you look at it. It's really, really cool. It's really hard to explain this interactive data piece without being able to physically see it. Okay, so that was my really quick overview of QIIME2. I tried to keep it to the 5-minute limit. I'm pretty sure I exceeded that. There's so much more to know, but it's just important that you remember that this is a bioinformatics program that can analyze large pools of data and then let you visualize it in really unique ways across the entire community of scientists and data scientists. So it just has really cool applications, and it's a really cool technique. I'm sure that we will see more from it, especially with AI learning coming on board. But that is all I have for you, so thank you for listening.